Hello students, this is your Prasanna ma'am from NEET World. Today I am here to deal with EAPCET PYQs from the chapter Cell Cycle and Cell Division. So if you look into the most recent question which is asked is of assertion-reason type, where in assertion he is saying like crossing over leads to genetic recombination. Yes, of course this is correct, and crossing over takes place in which phase beta? Crossing over takes place in pachytene stage. It takes place in pachytene stage of prophase 1 in meiosis. Pachytene stage of prophase 1 in meiosis. In reason, if you see crossing over is the exchange of chromatin bits between two sister chromatids. He is saying this is wrong. Crossing over always takes place between two non-sister chromatids of homologous chromosomes. What is the difference between sister and non-sister chromatids? What is a homologous chromosome actually means? See, in a homologous chromosomes, let us assume a trait like skin color. Skin color is a trait, let us assume. Okay beta, in skin color let us take two different alleles, one is capital D and one is small d. Capital D codes for melanin pigment which is dominant in D and recessive in small d. These two alleles are located at same loci on two non-sister chromatids of homologous chromosomes. They are located where beta? On two same loci of homologous chromosomes, non-sister chromatids of homologous chromosomes. So always crossing over takes place between non-sister chromatids. They will take place between non-sister chromatids and never between sister chromatids. These two are sister chromatids. These two are what sister chromatids. Crossing over never takes place between sister chromatids. Clear beta? So here if you see, assertion is false, assertion is true and reason is false, then our option will be option C. Next, the following type of cell division occurs in microspore mother cells to increase the number in microsporangium. Cell divisions in microspore mother cell. Microspore mother cell is a diploid cell, right? Microspore mother cell is a diploid cell. It always undergoes reductional division or meiosis. It always undergoes reductional division or meiosis to produce 4 microspores. To produce how many microspores 4 microspores. Yes, and that is how it is increasing in the number in the microsporangium. In microsporangium, it increases the number by meiotic division and releases out 4 microspores. So our answer is option 1. Next question, if you see, next question. Yes, here again assertion-reason type question appeared in the year 2023. Syncytium is result of karyokinesis not followed by cytokinesis. True. Syncytium means what Syncytium means multinucleated condition. Syncytium means multinucleated condition. Clear? So this occurs because of continuous nuclear division not followed by cytoplasmic division. So yes, assertion is true over here. In a plant cell, middle lamella formation starts at the center. Middle lamella formation starts at the center and grows outwards of the cell leads to cell wall formation. This grows outwards of the cell and this leads to what This leads to what? Cell wall formation. Wherein if you see, here it is middle lamella, Middle lamella is produced by what? Golgi bodies. Middle lamella is formed at the center because of the secretion of Golgi apparatus. This middle lamella turns to cell plate and moves outwards, and that is how two cells or the cytoplasm of the cell is separated, forming two cells. This pattern of cell division is called as centrifugal mode of cell division. What is it called as beta? Centrifugal mode of cell division. So yes, this is also true. But if you look into reason and assertion, nowhere reason is explaining the assertion or reason is linked to assertion. In that case, option B is that we choose, where A and R are true but R is not the correct explanation of A. Next question, if you see, cell that do not divide further due to the following changes. When does a cell will not divide further or it escapes the cell division? Just have a gist on the cell cycle, wherein cell cycle has two phases. What are they, Nana? Interphase and M phase. Interphase and M phase. Interphase is further divided into G1S and G2 phases. Okay, G1S and G2 phases. If there are few cells which are not very often dividing, for example a mature RBC cell or a differentiated muscle cell or a nerve cell, these cells will not divide very often. They will escape from G1 phase to G0 phase, and whenever they want to resume back their cell division or cell cycle from G1, they will enter into G0 and they will again come back to G1 phase. This is what the flow they follow by the cells which do not divide very often. M phase has two substages. One is karyokinesis and the other is cytokinesis Karyokinesis has substages: prophase, metaphase, anaphase and telophase, followed by cytokinesis. Followed by what, Nana? Cytokinesis. So if you look into here, a cell which will not divide will exit from G1 phase and they will enter G0 phase of the cell cycle. This is the correct option. After G1 it is entering S means it is a normal cell cycle. After G0 it is entering G1 means it is resuming back the cell cycle. Exit interphase and enter M phase is a normal cell cycle. So what is our option? Option C. Clear, ma? Next. Identify the meiotic stage in which homologous chromosomes separate while the sister chromatids remain associated at the centromeres. If you look into metaphase 1 of meiosis 1, you can observe two chromosomes forming bimetaphasic plate. What do you see, beta? Two chromosomes forming bimetaphasic plate. Bimetaphasic plate. So here during anaphase 1 without centromere splitting, this chromosome will move towards the upper pole and this chromosome will move towards the down pole, wherein two sister chromatids of the chromosome will move towards their respective poles. Clear, beta? Two sister chromatids of a particular chromosome will move towards their respective poles. So here this happens in which phase? During anaphase 1. Option C is the correct answer. Next question is match the following type, which is appeared during 2023 only, wherein if you see crossing over occurs in packetine, of course. Option 4, synapsis or synaptonemal complex formation, wherein homologous chromosomes are formed during zygotine, that is option 3. Weakening of synaptonemal complex or dissolution of synaptonemal complex, which is one and the same. This happens during diplotin. Terminalization of chiasmata is observed during the last phase of prophase 1 during meiosis, that is diakinesis. So if you see, A is 4, B is 3, C is 2 and D is 1. Option A is the correct answer. Next question. Again match the following type, appeared in 2022 from this chapter. If you look into it keenly, separation of homologous chromosomes. Homologous chromosomes get separated during anaphase 1, wherein sister chromatids remain same. Initiation of chromosomal pairing is called as what, ma? Synapsis. Chromosomal clustering at opposite spindle poles happens during telophase, the last phase of karyokinesis. Disc shaped part of centromere is kinetochore. Each centromere will have two disc shaped structures called kinetochores, which attaches to spindle fibers. This attaches to what beta? Spindle fibers. So yes, what is our option ma? A is 3, B is 4, C is 1 and D is 2. Option B is the correct answer. Next question, if you see, the amount of DNA in the cell undergoing mitotic division is doubled during S phase. Of course, DNA duplication or replication happens in S phase and doubling of DNA content happens in S phase. Assertion is true. It happens due to doubling of chromosome number in S phase of interphase. Correct? It happens due to doubling of chromosome number, he is saying. Wrong. Okay. Always do remember, it is DNA content that doubles. DNA content doubles, not chromosome number. If you look into chromosome in G1 phase, actually chromosome formation takes place in prophase ending and clear chromosome structure you will see in metaphase, but then for your understanding, if we take in G1 phase, this is G1 phase, okay, 2 chromosomes are there and let us assume 2 chromosomes are there and let us assume 2C is the DNA content. So this moves to after DNA duplication. Here also in S phase you will see 2 chromosomes, but the DNA content is 4C. DNA content is what beta? 4C. So yes, chromosomal doubling doesn't take place. Reason is false. When A is true and R is false, then our option is C. Clear? Next question. If an onion root tip cell has 14 chromosomes and DNA content as 2C. How many chromosomes? 14 chromosomes and DNA content is 2C. Okay. And the number of chromosomes and DNA content in G1, S and G2 he is asking. In G1 it is same chromosome number. In S phase of course DNA doubling takes place, but then chromosome number remains same. In G2 phase also chromosome number remains same and this doubled DNA will be as it is in G2 phase. Now what will be our answer? Option A, 14-2C, 14-4C in S, 14-4C in S and 14-4C of DNA content in G2 phase. Clear beta? Next question of 2021, if you see, fill in the blanks he is asking. During metaphase, spindle fibers attached to the disc shaped structures which are present on the centromere, those are what ma? Kinetochores. Those are what? Kinetochores. The disc shaped structures over the centromere. These are disc shaped structures over the centromere. To here the spindle fibers are attached connecting to the asters. Next, centromere split and chromatids separate during anaphase. It happens during anaphase of mitosis and anaphase 2 of meiosis. Anaphase 2 of meiosis. Clear beta? So here what is our option? Option C, wherein here it is kinetochore and they separate in the anaphase of mitosis, anaphase 2 of meiosis. Next question. Choose the incorrect statement among the following. Always whenever the statement based question is given, look into it properly whether he is asking you for a correct statement or an incorrect statement. Okay? Yes. So A, statement A says that cells of the lining of the gut are replaced by mitotic division. Yes, of course this is correct because these are what ma? These are somatic cells. These are what? somatic cells. Somatic cells always undergo mitosis, germ cells will undergo meiosis. Formation of lipid endosperm takes place of if karyokinesis is not followed by cytokinesis. 100% true, because karyokinesis, that is nuclear division, if it is not followed by cytoplasmic division, then multinucleated condition is observed. What is observed? Multinucleated condition. This is called as syncytium. True. Yes. And that leads to the formation of liquid endosperm. This you can see in coconut, that is coconut water. In telophase, the chromosomes reach the poles and start to condense to form chromatin network. No, they will uncoil to form the chromatin network. They will uncoil to form the chromatic network. Condensation, whenever this term comes, beta, do remember and keep it in your mind. Your answer should be prophase. Condensation always happens in prophase, uncoil during the last phase of karyokinesis, that is telophase. In quiescent stage, that is G0 phase, the cell is metabolically inactive. No, the cell is metabolically active, but it will not divide. That's it. So here, yes, this option is wrong. C is wrong. He asked us to identify incorrect statement. No, then option C and D we will choose. C is the correct answer. Next, choose the correct statements among the following. Correct statements here he is asking. Yes, so if you choose the correct statements, the stage between two meiotic divisions is interphase. Wrong. The stage between two meiotic divisions is interkinesis. It is what, Nana? Interkinesis. Next, during zygotine, pairing of homologous chromosomes and synapses takes place. 100% true. Initiation of mitotic spindle takes place during metaphase. Initiation of mitotic spindle takes place during metaphase. No, initiation of mitotic spindle takes place at the end of prophase only, at the end of prophase or transition to the metaphase. At that time only it will take place. In animals, mitotic division takes place in a diploid somatic cell and not in haploid cells. 100% true. So here what is our option? B and D are the correct answers. Option D is our answer. Next question. The chromosome number of a potato gamete is equal to diploid the number of meiocytes. Meiocyte diploid is 2n diploid. Potato gamete he is asking, that is haploid, of housefly, and thrice the number of meiocytes of a fruit fly. Okay, the meiocyte of a fruit fly has 8 chromosome number. Beta, what is the meiocyte of a fruit fly is having? 8 is the chromosome number. He is saying thrice. 8 into 3, that is 24 haploid gametes of a potato. Next, if you take meiocyte, it is double the meiocyte of housefly is saying. For housefly, for meiocyte it is 12. Chromosome number is 12. This is for fruit fly, that is Drosophila, and this is for housefly. Housefly. These are what, Nana? These are meiocyte chromosome number. Meiocyte chromosome number. Double he said, na, 12 into 2, that is 24, again haploid. We got the answer. But then what he is asking? He is asking to identify the haploid ratio of chromosome number of fruit fly, housefly and potato. Potato to we got, that is 24. Chromosome number, haploid and diploid meaning is what? Haploid means half the set of chromosomes of diploid number, right? Half the set of chromosome number of fruit fly. 8 by 2, half the set of chromosome number is 4. Half the set of chromosome number 12 by 2, it is 6. So what is the answer? 4 is to fruit fly to 4 is to 6 is to 24 is the answer. It is easy to do, but then little understanding is required, beta. It is easy to do only, don't get scared looking into such questions. It is easy to do, but then little understanding is required on what to do. Here yes, what will be our answer? 4:6:24 is the answer. Next question, if you see, identify the correct sequence of subphases during prophase 1 of meiosis. He is asking prophase 1 of meiosis. It starts with leptotene, followed by zygotene, then pachytene, then it is diplotene, then finally it is diakinesis. So what is our option? 3, then 1, then it is 4, then it is 2, lastly it is 5. So option C is the correct answer. Next question, ma. Arrange the following statements on meiosis in a correct sequence. Meiosis. So here if you see, yes, yeah. Firstly, pairing of homologous chromosomes. This happens during zygotene. Pairing of homologous chromosomes happen during which phase? Zygotene. Next, it is the appearance of recombination nodules. Happens during pachytene. Appearance of recombination nodules in pachytene. Then, dissolution of synaptonemal complex. Dissolution of what? Synaptonemal complex happens in diplotene. Terminalization of chiasmata, that is last phase, diakinesis. Next, homologous chromosome separation, anaphase 1. Lastly, disappearance of nuclear envelope, it is in telophase. Disappearance of nuclear envelope, yes. Yes. So all these activities are of what beta? Prophase 1. First it is yes zygotene, then pachytene, then it is diplotene, followed by diakinesis, then of course telophase. After that, only after prophase, then undergoes anaphase 1. Yes. So yes, it is B. Option, correct option is what, ma? B. Yes. Meiosis is characterized by which of the following? Meiosis is characterized by which of the following is he is asking? 2 nuclear divisions with 1 chromosomal dividing. Chromosome divides only one time. 2 nuclear divisions with chromosome dividing only how many times? One time. That chromosomal division happens during anaphase 1. Clear, ma? Next question. Next question appeared in 2019. The chromosomes align at the equator and the microtubules from opposite poles of the spindle get attached to the kinetochores of sister chromatids. Chromosomes align in the equator and microtubules from opposite poles of the spindle. Chromosomes align at the equator and microtubules from the opposite poles of the spindle gets attached to the kinetochores. They get attached to what, ma? Kinetochores of the sister chromatids. This happens in metaphase 1. In metaphase 1, it is what, ma? Bimetaphasic plate is seen, right? Bimetaphasic plate wherein 2 chromosomes align in 2 equatorial plates. This sister chromatid is aligned towards this spindle fiber, the kinetochore attached to this spindle fiber. Here it is attached to the lower spindle fiber. Clear, ma? So here the answer is A, metaphase 1. Next question. Identify the correct combination of statements. During leptotene, chromosomes gradually visible. True. Next, nuclear membrane and nucleolus do not reappear in telophase 1, they reappear. Anaphase 2 does not begin with simultaneous splitting of centromere of each chromosome in meiosis 2. Anaphase 2 does not begin with simultaneous splitting of centromere of each chromosome in meiosis 2, he is saying. No, they begin. Do not begin is wrong. They begin simultaneously with splitting of centromere of each chromosome in meiosis II. So yes, this is wrong. Next, prophase 1 is characterized by the appearance of bivalent chromosomes as tetrads in meiosis I. Prophase 1 is characterized by what beta? Appearance of bivalent chromosomes as tetrads in meiosis I. This happens during zygotine. True. So what will be our answer? 1 and 4. Option C is the correct answer. Next question. Again, assertion reason type appeared in 2019. In crossing over, the exchange of genetic material between homologous chromosomes occurs both in mitotic and no. Recombination never occurs in mitosis. The process of crossing over is only possible by mediated by recombinase enzyme. Yes, true. During packaging, recombinase enzyme during packaging. Yes, here reason is true, assertion is false. Then what will be our answer? Reason is true, assertion is false means D. Next question, ma. Match the following type in 2018. So condensation phase actually is the interval between M phase and S phase. Of course, our option is 3, that is G1 phase. Cytoplasmic condensation takes place, and as most of the cell organelle duplication occurs, condensation also occurs, cytoplasmic condensation occurs in G1 phase. In M phase though, condensation always our option is prophase. Decondensation phase, in which synthesis proteins takes place preparing for mitosis. After DNA duplication, proteins whichever are required for mitosis are synthesized in G2 phase, then our option is 4. Next, replication in which the amount of DNA per cell doubles. Replication in which amount of DNA per cell doubles during S phase, that is option 2. Next, quiescent stage of inactive stage. Inactive stage is interphase. Quiescent stage is no G0 phase. So G0 phase, it is option 5. G1 it is option 3. Then the flow of options is A3, B4, C2 and G0, that is 5. Option A is the correct answer. I hope you are getting the point, children. Next, match the following type of question again in the year 2018. Packaging, wherein recombination nodule is visible. Zygotine, pairing of chromosomes, that is 3. Diplotene, wherein dissolution of synaptonemal complex, that is 4. Leptotene, compaction of chromosomes. Compaction of chromosomes happens where ma? In leptotene, that is option 1. So if you look into the options 5, 3, 4 and 1, D is the correct answer. Next question, ma. Last and final question of the year 2018. Assertion reason based, focus on it. Assertion wherein meiosis increases the genetic variability in the population of organisms from one generation to the next, and such variation are important for the process of evolution. True. Errors in DNA replication is the main cause of genetic variability. Wrong. Errors during the DNA replication lead to what, ma? Lead to gene mutation, not variation. They will lead to gene mutation. So yes, reason is false, assertion is true. In that case, our option is C. In the next session, students, we are coming up with NEET previous year questions which are very important. Stay tuned. Thank you.